Hello, and welcome to night 4 of our 17-part series, An Evening with AP Biology. This evening, we will be talking about cellular respiration and photosynthesis. Last night, we mentioned some cellular components, your chloroplasts for photosynthesis and your mitochondria for cellular respiration. But we have to, of course, start our story with the formula and then talk about how prokaryotic cells can do respiration without mitochondria, because they do not have membrane-bound organelles. So cell respiration, true aerobic cellular respiration, happens in the mitochondria and does require oxygen. So your standard formula is glucose, which you're going to get from food, plus the oxygen, which you're going to get when you breathe in air, is going to release CO2, water, and all important usable cellular energy called ATP. But let's start our story off simple. Let's start our story with yeast and bacteria, and these cells are going to be able to do respiration called anaerobic respiration without oxygen. So if we look here to the right, we're going to see this process called glycolysis. There, you're going to take glucose, and you're going to have a intermediate and end up with either ethanol in alcohol fermentation or lactate in lactic acid fermentation. Both of these do not require oxygen and will yield 2 ATPs and 2 NADH molecules. You can see here in another simple form that, well, in both cases, you can see that the NADH that's formed is going to lose its hydrogen to NAD+, which will cycle back around in both alcohol and lactic acid fermentation. Aerobic respiration is what we will do, and it requires oxygen. It will happen 2 places, cytoplasm and mitochondria. And once again, oxygen is present. There will be an initial investment phase with 2 ATP molecules, but we'll have a greater yield once that process is done. And if all is good in the world, a maximum of somewhere between 36 and 38 ATP molecules will be produced. So the first stage is going to be, just like it is in bacteria, glycolysis. And that's going to give you a few potential ATPs, because remember, it's going to give you natural ATPs and it's going to give you these molecules called NADH, which we will see how they work in a little bit. You're then going to move into the matrix of the mitochondria, where the citric acid cycle is going to produce many more NADHs and FADH2 molecules and ATP. And then the electron transport chain, which is going to happen in the inner mitochondrial membrane, is where all of these ATPs will eventually be produced. And we'll see how that goes very, very, very shortly. So most of the NADH and FADH2 are going to be generated in the matrix in what is known as the Krebs cycle. CO2 will also be generated along the way, and that will be released. Now, there are many intermediates here, but don't get hung up in the intermediates, and don't even get hung up where the NADHs and where the CO2s are being made. Just know that NADH is being produced in several places. FADH2 is being produced, and you are, along the way, releasing CO2. You're also producing GTP, or another energy carrying equivalent to ATP, here also in the Krebs cycle. So most of the ATP molecules are going to happen when NADH or that FADH2 molecule donate their electrons in this electron transport chain. Now, this is located in the inner mitochondrial membrane, and it's going to help create what's known as a hydrogen ion concentration. This hydrogen ion concentration is extremely important because it's going to set up this proton gradient, and that's going to allow these hydrogen ions, or these proteins, to travel very fast through a channel protein called ATP synthase. So let's see what happens. The electron transport chain is a series of proteins that are embedded in the inner mitochondrial membrane. And if you notice, let's kind of watch what happens. Let's get rid of this here. The NADH here and the FADH2 here are going to donate their electrons, which have a tremendous amount of energy. That energy is going to be enough to pump the hydrogen ions that are already in the matrix against their concentration gradient. So very much like active transport, where ATP is used to pump things against their concentration gradient. Here you're pumping hydrogen ions against their concentration gradient. Now that's going to cause them to build up in what is known as the intermembrane space. And the intermembrane space is going to be found in between. If we kind of look at a mitochondria, it's going to look something like this. That's your outer mitochondrial membrane. There's your inner mitochondrial membrane. This here is the matrix. So this is the matrix right there. And this is taking place right on that inner mitochondrial membrane. So this space right here is your intermembrane space. Now what's happening is the hydrogen ions are getting pumped in there. Now by doing so, let's get rid of this so we can see, you have a tremendous buildup. This is going to make what is known as proton motive force. Now, because of that, these hydrogen ions can now move down their concentration gradient through this protein channel called ATP synthase. Now, they're going to come through with a lot of energy. They're going to come through with enough energy to take an ADP and take that inorganic phosphate right there and bind it to the ADP to make what is known as an ATP. Now, the reason why they say NADH can net you 3 ATPs is because you'll see that NADH happens early, and you're going to go through 1, 2, 3 of these proton pumps. FADH2 joins the party a little bit later and only goes through 2 of those pumps. And that's why FADH2, they say, will net you 2 ATPs, where NADH will get you 3. So this electron transport chain is going to pump the hydrogens across into the intermembrane space, then allow them to come screaming back in through the ATP synthase, generating a lot of ATP. Now, this is all possible because of this oxygen. This oxygen right there is what is known as an electron acceptor. So because these electrons, after they go, we'll call it whizzing through the electron transport chain, they're going to come back around, and it's the draw of the oxygen that's pulling those electrons that allows the energy to pump the hydrogens out of the matrix into the intermembrane space. So without oxygen, none of this happens. That is the major difference between aerobic and anaerobic respiration. Now, what about photosynthesis? Now, photosynthesis is occurring only in plants. They're going to be called autotrophic eukaryotes. What that means is they do have membrane-bound organelles, and these membrane-bound organelles are called chloroplasts. And what they're going to use is CO2 that's in the atmosphere, water that they have to get from the ground. They're going to pull it up through their roots with the help of solar energy. It's going to create glucose and oxygen. Now, there are 2 parts to the photosynthesis story. There is what are known as light-dependent reactions, and that's going to happen in the thylakoids, and that's where the water is going to come in. There's your water right there. The sun, and with the help of enzymes, is going to split that water. It's going to release the O2. We know that chloroplasts release the O2, and so do plants, which is going to create a little bit of ATP and this molecule called NADPH, very similar to NADH, but a little different. That's going to move on. And what's going to move on to the stroma, which are known as the dark reactions or light-independent reactions. They do not happen at night. They just do not require the sun directly. They're going to enter into something called the Calvin cycle. And in that, you're going to take in the CO2 from the atmosphere, which we just talked about, and you're going to create what is called the G3P molecule, which is going to ultimately go on to form glucose. Now, the stroma is this area that's in the chloroplast, but that's not in the thylakoid. So there are many stacks of these thylakoid over here. There's just one here, but there are many. So the stroma is the area that's not in the thylakoid. Photosynthesis, once again, is broken down into two portions. I'm going to call it the light reactions, which do require the sun, and happens in the thylakoid membrane, and does require solar energy, which we just said. The water is taken in and it is broken in a process called photolysis, and with the help of the sun, but enzymes are needed, so please don't think it's only the sun. That is going to allow for a lot of hydrogens to be around. Well, we're going to have, again, an electron transport chain where it's going to be set up with the electrons from the water. So what happens here is there's your ETC, or your electron transport chain. Now here they're going to be called photosystem 2 and photosystem 1. And what's going to happen is light's going to come in at either P680, which is nanometers, or P700, which also are nanometers, which is going to allow, and if you look at photosystem 2, which actually is first in the line here, that's where your water is going to be split. Now your electrons are kind of excited, which are then sent down through this electron transport chain, and look what happens. You're going to pump hydrogens. You're going to pump them against the concentration gradient. Here, however, you are pumping them into the interior of the thylakoid, known as the lumen. Again, you will have what is known as proton motive force, and that is going to be a tremendous buildup of hydrogens, which will then go down their concentration gradient through ATP synthase, allowing you to generate ATP. Now, plants do not generate as much ATP as the mitochondria do, but they do it. Also, do not forget, plants do have mitochondria, and they can make ATP through aerobic cellular respiration. The act of moving these hydrogens down their concentration gradient by this proton motive force is called chemiosmosis, so you may see that word. And that's simply moving of the hydrogens down their concentration gradient through the ATP synthase. The reason why they can be moved down is because they were built up with the help of a pump and the energy from the electrons. Now, a plant does need more ATP than it does NADPH. That might be a problem. So in order to kind of help that situation, a plant may go through what is known as cyclic photophosphorylation. And in that case, what's going to happen is the electrons are going to get excited, and then they kind of cycle back around. They do not know NADPH is produced here. Because they cycle around, you're going to continually only generate ATP. Once the plant has enough ATP, it can then go back to what is known as your non-cyclic, or what we talked about earlier, which is your normal movement of electrons, which are going to go through both photosystem 2 and photosystem 1. And there's your NADPH being produced. Okay, so some ATP will form by this process, and that ATP will be used in the Calvin cycle. In addition to the movement of the H plus ions, this NADPH is created, and the Calvin cycle, as we said, happens in the stroma. So here's another version of that same picture. There is your chloroplast. You can see that's the stroma. These guys are your thylakoids. And you see here, water comes in, oxygen leaves. You split the water by a process known as photolysis. Two things are created, ATP and NADPH, which are going to go off to the Calvin cycle. In the event, once again, you need more ATP than NADPH, the plant will go into what's called cyclic photophosphorylation. That ATP and NADPH will enter into the Calvin cycle, taking in CO2 from the atmosphere and ultimately producing a sugar. This only happens during the day. even though it is light dependent and light independent, the whole process only happens during the day. Please, please, please understand that. No photosynthesis at night. If we take a look at the Calvin cycle, once again, Calvin happens in the stroma. There is, again, a lot going on here, and you do require some ATP. If you notice, you need 6 ATP here, and you need 3 ATP here. That's 9. You need 6 NADPHs. That means you do need more ATP than NADPH. That is why, once again, a plant may have to go through cyclic photophosphorylation. Probably the most important enzyme on the planet is that one right there. It's called rubisco, and that's what's going to allow this carbon dioxide to enter the Calvin cycle and be incorporated into these, sugars, if you will. It happens in what's known as the carbon fixation phase. Then we're going to do a little reduction, and that comes from the electrons. And we have to regenerate our ribulose bisphosphate. Again, that's this right over here. Each time you go through the cycle, you will go release 1 G3P, which will again eventually go on to make glucose. So, in summary, we know that the mitochondria is where aerobic cellular respiration will happen in both plants and animals. In the event that something like a prokaryotic bacteria does not have mitochondria, it will only do glycolysis, but they don't really need that much energy anyway, and they're pretty small. If you do aerobic cellular respiration, you will net somewhere between 36 and 38 ATPs if all is perfect. It will be done through this glycolysis phase, which happens first, and then ultimately entering the matrix and the Krebs cycle. If you are a plant, you are going to have chloroplasts, and you will use the sun, and you will use water. And with the help of carbon dioxide, water, and the sun, you will generate glucose and oxygen as a byproduct. So hopefully that was helpful, and we'll see you tomorrow night.